Hi, everybody. Welcome to ResearchCon 2020. My name is James Lawler. I am an adult with CF, and I am one of your co-chairs for this event. We have a really fantastic agenda today that you're going to hear more about in a bit. But first, I'd like to welcome my fellow co-chair, John Sheridan.
Hi, everyone. I'm John Sheridan. I'm a research director at the Cystic Fibrosis Foundation, and I want to first thank everyone for joining us. As James said, we have a really great agenda for everyone.
So once again, my name is James Lawler. I am one of the co-chairs this year. I'm 33 with CF. I live in Huntsville, Alabama, and I've been active, of course, with the ResearchCon and the Adult Advisory Council. And in the rest of my time, I am really fortunate to work as a computational biologist in the field of pediatric genetics research, in addition to all of my advocacy. So I'm really excited about ResearchCon, mostly because I'm a huge nerd. And it turns out that cultivating an interest in learning about CF research and understanding how CF works has really helped me cope so much as a patient because it lets me address a lot of the uncertainty that comes with CF. I mean, sometimes things go wrong, you have setbacks, your health doesn't act the way that you want it to. And for me, it really helps to be able to understand what's going on, to be able to understand what other new treatments might be coming in the future. And it really helps me to have a language of science and research that I can use to talk to my care team in a whole new way. And it helps me have new and different and more productive conversations. So one plug for one of our events we have this evening is CF 101, where we're going to introduce some of that language. So check that out if you're interested. The other thing I love about ResearchCon is that it brings researchers and patients and families together into the same event. It's for the entire CF community, and it actually lets patients like me and researchers like John connect with each other and talk about the things that are going on in the field. And that means it's a really safe and important place for us to ask questions. And that's why we have the Q&A features that you'll hear a little bit more about. So with that, John, please tell us about yourself.
Sure. James, first I just want to say it has been a real pleasure serving as the co-chair with you on ResearchCon this year. It's been a wonderful experience. So my name's John Sheridan. I am a research director here at the CF Foundation, and I'm really excited to be here with you at ResearchCon this year. So the focus of this year's event is on the next generation of CFTR research and genetic-based therapies. So I do first want to thank all of the speakers and volunteers who have really contributed a lot of time and effort to really making this year's ResearchCon so wonderful. So each information session has been developed and shaped by the CF community, and we hope you'll gain new insight into the biology of CF, the scientific research, and how new therapies targeting CFTR are impacting outcomes in the future.
I'm really excited for this keynote. We're going to start out with a presentation from Dr. Bill Skatch, who is the Executive Vice President and Chief Scientific Officer of the CF Foundation. And then I'm going to lead us in a panel conversation with Dr. Skatch as well as Dara Reba, who is both the Director of Clinical Research Awards at the CFF, and she is also a member of the CF community because she is a mother of 2 children with CF. So like I said, this is a safe space to ask questions and share your experiences, but I do want us all to keep in mind a couple of pretty simple ground rules. One is to always respect your fellow ResearchCon attendees and their own life experiences. And then also that please keep in mind that your questions are open to everyone to see, and that we can't address anybody's particular medical situation. So please keep your questions as general as you can so that they'll be able to help everyone. But definitely do ask questions. So I'm going to quickly review the technology of how we can all interact. Just like we did for the COVID section, we will be taking audience questions during the session, so you can submit those at any time by clicking on the right side of your screen that's labeled Q&A. You can also see other questions there. Give a thumbs up if you want a question to be answered. I think you guys all got the hang of that really well in the first place. And we'll do our best to answer as many questions as we can. You'll also notice that there's a chat bubble icon on the right side of your screen. That is the group chat for everyone in the session that lots of folks are already using. You can open that if you like and participate, or you can hide it if you just want to focus on the conversation. And then if you have any technology problems during the panel, please go back out to the environment and go to the tech and program support room on the home screen of the event that looks like a lab. Also, check out the resources room. You'll find several helpful resources there that have been compiled from all of the different sessions. So there's lots of cool stuff. One thing that I wanted to point out that I hope will be useful to a lot of folks is the CF 101 glossary, where we have gone and tried to give kind of normal language definitions of some of these different terms that you're going to hear in ResearchCon. So Bill, why don't you John, why don't you introduce Bill for us?
Sure. Thanks a lot, James. As James mentioned, our keynote speaker for this year's ResearchCon is Dr. Bill Skach. He's the Executive Vice President and Chief Scientific Officer of the Cystic Fibrosis Foundation. So Bill joined the foundation about 6 years ago, and in his role, he oversees all academic and industry research programs supported by the CFF. Prior to joining the foundation, Bill had a long academic research career where he studied the processes and mechanisms governing protein biogenesis, which includes the folding mechanisms of CFTR protein. And some of you may be aware, but late last year at the North American CF Conference, Bill announced the Path to a Cure initiative. And this is the foundation's ambitious research program to support development of treatments for the underlying cause of disease and ultimately a cure for CF. So during his keynote address, the 3 R's of CFTR, restore, repair, replace, he'll be giving an overview of the Path to a Cure initiative and some specific challenges that need to be addressed to develop a curative therapy. So please give a virtual welcome to Dr. Bill Skach.
All right. Well, welcome everyone. I'm absolutely delighted to be here tonight. And although COVID-19 has really dominated much of our thoughts recently, we're here to tell you that our mission to cure CF has not changed. We are as committed as ever to continuing to move forward. We are looking for the best science. We're recruiting the best scientists. We're bringing in the best companies. But we are still operating on all cylinders. And so there's some very, very exciting things going on that I'm very pleased to have a chance to talk to you about. So over the next 15 minutes or so, I'm going to put a presentation, which, as James said, will be followed by a panel discussion. So to kind of overview the talk, we're going to talk about the path to a cure and really explain what this is, why we're doing it, what it means for all of you. And the goal up front is really to treat the basic defect in 100% of people with CF and ultimately to develop a cure. That is kind of the fundamental process that we're working on. The CF Foundation has really been trying to do this ever since it was started. And the path to a cure is really now a transition from where we were in our therapies and our treatments for CF, and now a transition into new areas that are going to have completely different and much more exciting outcomes. We've learned a lot from modulators, and some of the lessons from CFTR modulators we can take directly into help us with the path to a cure. And we'll talk a little bit about that tonight. Why is this the right direction and the right time for CF? Well, we were in this gene therapy space 20, 25 years ago, and we were way out in front of the curve, probably a little further out than we should have been, because at that point the science was not yet developed. What's happened in the interim is the gene therapy space, the genetic therapy space has exploded in other areas, other diseases, new scientific discoveries, and new technologies that now make it the perfect time for CFF to really get into this space. And as such, we're really entering a new area of genetic therapies. And that means that therapies will be tailored to specific mutations as opposed to general symptoms. And I'll talk a little bit more about that in a moment. We've learned from modulators that not all therapies will treat all mutations. And so we need new strategies. We need new approaches that we haven't had before to really address the problem for everyone. And some of these therapies we'll be talking about involve messenger RNA, DNA or gene therapy, gene editing. And one of the things we've learned as we've started the path to a cure is that this requires a new language and really a new explanation so people can understand what it is we're really trying to do. So the path to a cure is really an ambitious agenda, probably one of the most ambitious agendas the CF Foundation has ever undertaken, to treat the underlying cause of CF in everyone, 100% of people, and ultimately to deliver a genetic cure. We've allocated $500 million to this initiative, which is the largest allocation we've ever made for the first 6 years of this project, to really get this off the ground. And the goal here of making this announcement and putting these resources forward is really to challenge the academic community to come up with new science, to come up with new discoveries, to challenge industry to come up with new ways of designing therapies, designing treatments, and testing those treatments as we go forward. We will fund in this process all of the sound scientific programs that come to us. And that, I think, is really going to be allowed by this $500 million initial investment. So we want to make sure that we don't leave any stones unturned if there could potentially be something valuable underneath them. And if during that time it requires more resources, we are willing to put those resources forward. This is our eye of the bullseye. This is now the primary goal of our research programs, and that's why I'm so excited to talk to you about it tonight. So just to kind of outline this, where do we start? Well, we start with the mutated CFTR gene. That's the basic underlying defect that everyone with CF inherits. 2 copies of the CFTR gene. In people with CF, both copies have a mutation, and we need to find ways to treat both of those copies. And what do we need to figure out? Well, there are 3 basic buckets. Repair the CFTR protein and restore the CFTR protein when none exists. And I'll talk about each of these in turn. But we think that these 2 strategies will provide treatment for 100% of people as quickly as possible. And the reason we're going after those 2 is that that's our goal, is to really hit 100% so that we can restore the basic functional process of CFTR in everyone as quickly as possible. The 3rd bucket is really to fix or replace the CFTR gene. And that's what we mean when we start talking about a cure. Fixing the gene means that the basic genetic defect will no longer exist in the tissues that are affected in CF. And so the goal is now 2-pronged, to work as quickly as possible to bring therapies to 100% of people. And ultimately, while we're doing that, to work to develop a cure where people will no longer have the genetic defect in the first place. So I'm going to talk about 4 basic strategies. In order to really understand what we're doing, we need to think a little bit about the biology. And this is a slide that you'll probably see later in the evening that talks about kind of how we've been approaching CF and what our new thoughts are as we move forward. So the biology tells us that the genetic material, the memory of all of us, of who we are and what we're made of biologically, is in the genes or the DNA. And in the case of CF, there are mutations in that DNA that are inherited. Well, the biology shows us that the gene is basically a template. It's a memory, and it's used to make RNAs. and we'll talk a bit about messenger RNA. Messenger RNA is a copy. It's copied from the gene, and it's the transition state to go from the gene to making protein. Think of it as an intermediate. RNA is used to make protein, and proteins are the workhorses of the cell. They do things like make your muscles move and make your heart beat and pump fluid out of the kidneys to clear the blood and filter the blood with liver. All the proteins in the body have tasks to do, and they're all encoded by information in the DNA. So if you inherit a mutation in your DNA, that is copied into the RNA, and that mutation resides in the RNA. And the RNA is used to make protein, and the mutation then results in an abnormal protein, which in the case of CF and CFTR, mutated CFTR protein causes all the manifestations of CF. The lung problems, the liver problems, the pancreas problems, the GI problems are all caused because there is a mutation that's been carried into that CFTR protein and it doesn't function properly. So if we look at therapies to date, the last 50 years, almost all of our therapies have been targeted at CF manifestations. Mucus clearance, mucolytics, nutritional supplements, antimicrobials are all treating the end stage of this cause in CF manifestations. And we're only now just starting to work on protein-based therapies. And those are the CFTR modulators that can actually repair the CFTR protein to make it work. But if we move back further in this paradigm and we look at the RNA, this is an area that we have not yet approached. And if we go even deeper, the gene, that's where the fundamental defect lies. That's another area that provides a new opportunity for therapies. And if we look at the path to a cure, the path to a cure really spans now the gene, the RNA, and the protein to try to treat the basic defect that causes the manifestations of CF. And so I'm going to walk through each one of these areas, the gene, the RNA, and the protein, to let you know what we're thinking about, how we're planning to move forward, what types of strategies we're taking. And I think you'll agree that this is an extraordinarily exciting time because up until now, we really haven't had the tools to work at the RNA level or the gene level. And in recent years, that has changed completely. And now there are many tools coming forward. There's exciting discoveries in the laboratories now occurring on a weekly or monthly basis that are really changing the way we think about our ability to treat diseases like CF and CF in particular. So that brings us back to the 3 R's: repair, restore, and replace. Repair the broken protein. This is what we're talking about when we talk about CFTR modulators. Restore the protein when none exists. These are mutations that don't make any protein, and they basically revolve around RNA-based therapies. Molecules and therapies that will target the RNA molecules to make the RNA work better so we can make CFTR protein. Replace or repair the defective gene. That's now getting at the root cause so that we can now actually go after DNA-targeted therapy. So how are we going to do this? How do each of these play out as we go forward? For repair the broken protein, I think we're all fairly familiar with. This has emerged on the scene for the last 7-8 years. We have now seen a tremendous progress in moving modulators from a very small population to a much larger population, from modulators that don't work very well to modulators that work really pretty well. And we now have 38 mutations covered. About 90% of the US population carries those mutations. So we expect that when this moves into younger age groups, that we'll have about 90 to 91% of people in the US that could benefit from modulators. And we've already seen amazing responses in lung function, exacerbations, sinuses, GI symptoms, fertility. These have been really amazing. But we know that not all mutations will respond to modulators. We also know that not everyone tolerates modulators. So we're not yet finished. The modulator story is just getting started. It's being written as we're speaking. What are we doing in the future? Well, one of the things we want to do is to continue to expand modulators to cover more mutations. We know that there are a lot more mutations than the 38 that are approved that will respond to modulators in the lab. And so there's a process called theratyping where we can look at individual mutations in the laboratory and test whether or not the drugs, Trikafta, Symdeko or Canby, Kalydeco, actually work on those particular mutations. And the CF Foundation is testing 650 of these mutations that we think have a chance to respond, and we think that this will help move those forward. We're working with Vertex. It's also doing their own theratyping process. Vertex is now looking at a smaller number, but working with an assay that the FDA has recognized as an important assay for moving drugs on label. And we expect that sometime this year, there will be some effort by Vertex to move additional mutations onto the label. And we're watching very closely to see how that will play out with Vertex and the FDA and our data as well to identify which mutations might respond the best. We'd like to develop better modulators. Well, what does that mean? Well, modulators right now, if you look at Trikafta, it provides somewhere between 50 and 60, maybe 70% of normal CFTR activity. That is the normal protein. You take a broken protein like F508del or G551D, and the modulators work at about 50 or 60% effectiveness. We would like to find modulator combinations that work better than the triple or better than ivacaftor. We would like to return CFTR activity to normal. And we can see that in the sweat test. People that go on modulators can watch their sweats decrease, their sweat chlorides decrease, but they don't usually go back to normal. We would like to find modulators that actually do make it into the normal range. We also think it's important that we have new modulators so there's choice. If someone doesn't tolerate a modulator, if they have an allergic reaction, or if they don't respond well to it, we'd like to see additional modulators that could be tested, and so that physicians and patients could choose which one works the best for that individual person. So we think all of these reasons are why we should keep going forward with modulators. And I just want to mention that the CFFT lab is testing modulators from a variety of companies that are still looking at whether or not they work and trying to identify combinations of modulators that work as well or better than the modulators which are currently on the market. So this is very exciting. There's a lot more to come. Stay tuned. There's things going on behind the scenes, and I think in the next several years we'll start to see some of these programs advance. So restore the CFTR protein when none exists. And as I said, these are now RNA-targeted therapies primarily. We know there are mutations that simply don't allow CFTR protein to be made. And if there's no CFTR protein, there's no way you can repair that protein. One class of mutations is the nonsense mutations. And these are mutations like G542X, 1282X, 1162X. You can call them nonsense, you can call them premature stop mutations. They're basically mutations which insert a stop sign into the messenger RNA, so that as that messenger RNA is being translated into a protein, the stop sign prevents that process from happening. And what we're looking for are drugs which will read through the stop sign and enable the cell to make full-length protein. Now, many of you are familiar with the work that went into developing the drug called Adalurin. The CF Foundation was quite involved in that process, and we actually started these efforts over a decade ago. Unfortunately, Adalurin was not effective enough at reading through the stop sign to benefit people with CF. So currently, we're working to find better drugs, more potent, more active than Adalurin, that will actually now read through the stop sign with enough efficiency to make CFTR protein that will benefit, that will reverse the symptoms of the disease. We're funding basic science in this area with academic researchers. We have two very large screening assays that have together screened over 3 million compounds. Southern Research, Icogen are two very large screening facilities, and we're working with them. There is actually a clinical trial ongoing now with a molecule, a drug, which has the potential to read through these stop signs. That's being run by ELOX. It's being tested on people with G542X in the US and Europe, although it's been paused because of the COVID-19 issues. But we hope that that will give us an answer as to whether or not a drug that's more effective than Adalurin in the laboratory can be effective enough in the clinic and benefit people with CF. And our goal is to find out how much read-through of that stop sign do we really need to get in order to be a true treatment that benefits people. The CFFT lab is very active in this area, and they've been working with assays, making cell lines, transferring them to industry, and really working to try to help facilitate all the drug discovery efforts that we have ongoing. I want to say a word about splicing mutations. These are mutations that are somewhat complicated, but they basically affect mRNA processing. When mRNA is made, it's made in a very, very long chain. It gets cut into pieces, and then those pieces get stitched back together. And that's what we call mRNA splicing. And sometimes the pieces don't get stitched back together or cut in the right spot, so that when you end up with your mRNA, it can't make a protein. So if you don't stitch together properly or you don't cut properly, you have a processing defect. These mutations are like the 3849 plus 10 kb C to G, and they're very complicated names. But basically it means that there's a problem with the splicing and dicing of CFTR mRNA. One of the new technologies that's come out is that you can take very small snippets of RNA or DNA-like molecules that will hone in on the specific splicing sites, and they will convert an abnormal site to a normal site. And so you can re-stitch the RNA together and make a normal functioning messenger RNA, which makes a normal functioning protein. So very exciting work is going on at a company called SpliceSense that we funded earlier, and they're now moving forward in their preclinical studies. They're not yet in the clinic. They're trying to look at whether or not it's safe, whether or not it can be delivered properly. And so there's more to come in that, and we're very excited about that technology. And there's a whole series of other mutations. There's about 500 other mutations that include things like deletions, where large chunks of the gene are missing, so large chunks of the mRNA are just gone. There's frame shifts that make the mRNA unable to make protein, small insertions that also make the mRNA unable to make protein. And these are very complicated because they're very diverse. There's not one specific strategy that you can take. So there's no specific therapies for these other mutations. But what we're now looking at is a technique called mRNA replacement. And the good news is that mRNA replacement will not only treat these other mutations, but it will also treat splice mutations and nonsense mutations. In fact, mRNA replacement will treat 100% of all the mutations that we find in all people with CF, regardless of what mutation that person carries. So we're incredibly excited about mRNA replacement. Let me just tell you a little more about it. This is a technique where we take a normal copy of mRNA and we package it up in a tiny microscopic fatty droplet that we call a lipid nanoparticle. And basically, if you could imagine a microscopic particle of fat, and buried in that fat is a messenger RNA or multiple copies of messenger RNA. You can take that now in solution and aerosolize it, much like a usual nebulizer. You can inhale those compounds and then deliver the RNA straight to the epithelia of the lung. Right now, it's very difficult to deliver to the entire body, but we're going after the lung first with the hopes that we can now find new ways and more strategic ways to deliver to specific tissues. mRNA replacement will probably be made on a daily or every few days, or maybe a weekly, or even maybe every 2-week basis, but it's going to have to be repeated. And I will say, we now have a clinical trial ongoing that Translate Bio is sponsored, looking at mRNA in the clinic in people with CF to deliver CFTR mRNA. This is now looking at safety. It's looking at whether we can dose this once, dose this again. And we have backups. If Translate Bio is not successful, we're funding a company called Arcturus. They're in the laboratory testing their lipid nanoparticle or their fatty droplet recipe. We also have Arcturus, which is going to start testing additional lipid nanoparticles in 2020. Last topic is replace or repair the defective CFTR gene. And this is where we're now talking about DNA-targeted therapies. This involves gene transfer to deliver a normal copy of CFTR DNA to the cell, and gene editing, which takes the person's own DNA, which is already there, and fixes the mutation. Gene editing, you've probably heard about in the news, involves CRISPR. It involves techniques called base editing, zinc fingers. There are many ways now to do gene editing. But the exciting thing is, 5 years ago or 7 years ago, there was no gene editing around. It's really a newly invented technology that has just emerged on the scene. And there are many, many advances which are occurring constantly. These therapies enable a permanent correction. They enable a genetic cure, so that you won't have to take repeated therapies to restore or replace your CFTR function. We anticipate that these therapies will also start in the lung, but ultimately our goal is to develop ways to target all the tissues, the lungs, the gut, the liver, the pancreas, that are affected by CF, so that they will, again, have the normal CFTR gene and then develop normal CFTR mRNA and make normal CFTR protein. I want to just say that the genetic cure will treat the underlying defect. It will restore CFTR function, but it will probably not undo all the chronic damage in the lungs and the pancreas that has accumulated over years, and it will depend on how much scarring and how much irreversible tissue damage there is. So there will still be a need for some people for antimicrobials, for anti-inflammatories, even though their CFTR is functioning, just like we see now with modulators. What are the challenges? Well, we need to understand the basic biology. How can we actually accomplish this? Cells have evolved over millions and millions of years to keep DNA out of their cells and to make sure that their own DNA stays intact. We also need to identify the specific cells to target. We need to establish really clear and useful models to find out if the therapies actually work. And we need to develop testing facilities where companies can come to test their product to see whether or not it works in CF. We also need to advance these therapies into small clinical trials. We won't have thousands of people that can enroll in these trials. There may be dozens, hundreds, or even dozens. Currently, we're in discussion with about 15 groups, all interested in working on CF, all interested in taking their technologies and moving it forward. And we're really excited about that. We have several gene therapy programs now at 4D Molecular, which is based in California. It's Firevent, which is formerly Talibio. Abiona has a CF program that they've announced. We're waiting to see where that goes. We're not funding it now. And the UK Trust is planning their own next generation gene therapy program. So I'll finish the talk with this, research support. What we do at the foundation is to support research at any level that we think will advance the science. We fund basic researchers, and we've just started a new Path to a Cure research grants program where researchers can apply for funding to specifically cover the topics that I've just been discussing. We bring groups of scientists together into consortia, and we have them work on stem cells, on gene editing, on gene delivery, and on nonsense mutations, really challenging them to solve the difficult problems. And of course, the CFFT lab has been developing functional assays, model systems that they transfer to academic and industry laboratories, develop tools. They're doing gene editing in the lab right now. They have a lung stem cell biology, a program. There's bioinformatics and DNA sequencing, and they have a very large nonsense mutation effort. I should qualify that. The lab right now has been affected by the COVID, so the things at the lab have pretty much been put on hold. But as we go back to work and as we continue to recover from the COVID-19 distancing, all of these activities will be ongoing. So I'm going to stop there, and I'm going to turn it back over to Jim and stop sharing my screen. Okay, James, are we back on?
Yes. Yes, it took me a second to figure out that my video was on. So thank you very much, Bill, for that presentation. So now we are going to move into a panel discussion where we'll get to go into a little bit more depth on some of those topics, maybe answer a couple of questions, and really get a community perspective on some of the things that you just talked about. So definitely for those of you watching who thought that Bill's talk was really cool, like I did, you'll want to check out the Treating Rare and Nonsense Mutations section of ResearchCon, or the Genetic Therapies section, or really any of them, but those two in particular will go into more detail on some of the particular avenues of research that we heard about. And now I would like to welcome Dara Riva, who, like I mentioned earlier, has multiple connections to the community, both being a mom of 2 kids with CF and the director of Clinical Research Awards at the foundation. So Dara, welcome. Thank you for joining us. And please tell us a little bit about yourself and kind of what your journey with CF has been.
Hi, everybody. I'm really excited to be here. Thanks for inviting me to speak at the keynote tonight. So as James mentioned, I work at the Cystic Fibrosis Foundation. I joined in 2017, and I'm the director of Clinical Research Awards. So my day-to-day job at the foundation is really to accept proposals from researchers at care centers or institutes affiliated to care centers who are doing clinical research, and we have them evaluated. And then if it's good science and it is important to people with CF, then we will provide them with support, funding support to do their research. So that's my day-to-day job, and that's what I do at the foundation. But also, as James mentioned, I do have 2 children with cystic fibrosis. So my son is 10. We found out he had CF when he was born because he was born with meconium ileus. We had no idea that my husband and I were even carriers. And then I have my daughter, 8, who's 8. She is actually 8 going on 18. And they've been in the house for a month, so I'm surprised they're very quiet right now. My husband must be bribing them. She also has cystic fibrosis, and she was also born with meconium ileus. They have the F508del mutation, and then they also have the W1282X mutation. So we were really hopeful for Adelorn when that was coming through the clinical trials, and we were quite disappointed when that fell through, but we are very, very excited about Trikafta and the thoughts of that. So that's a little bit of my background.
Yeah, thanks for sharing that with us. I think that brings exactly one of the points I was touching on earlier, where kind of understanding the research that's going on really helps me deal with setbacks like that. And I'm sure you being closely involved probably felt the same way.
Yeah, absolutely. And I do get to the behind the scenes view of what happens with the CF Foundation and the research funding programs. And it does bring me a lot of hope and joy. There are some tough days because it's not always good news. And it's not always positive results that we see in clinical trials. But for the most part, I feel very blessed.
Great. So I'm going to start off with, I think, one of the big questions, which is, what do we mean when we say the word cure? And especially, I'm going to ask you guys to kind of contrast maybe what that means for an adult like me that's been walking around with CF for a few decades, and maybe what that means for someone younger. So let's start with Bill.
So James, that's a terrific question, and I think what I will do is talk more about it from a scientific approach. And I think Dara has probably a very different perspective, which may be more relevant, but I think both of them are really important. From a scientific approach, when we think of a cure, we think of what is the true basic defect and how can we overcome that basic defect. So to cure CF from a genetic perspective would be to really fix or replace the defect that is inherited, which would restore the normal gene, restore normal RNA synthesis and normal protein synthesis, and restore normal CFTR function in a way that the person would not need to take medications like a modulator every day of their life to make sure that CFTR is working properly. So from a scientific perspective, that would be, in our view, a cure. And if you're young, and if you have really normal lungs, and you get started early enough, even maybe to reverse some of the pancreatic damage, that would then be a cure, and people could live a completely normal life. As you get older, and as the tolls of CF are taken on tissues such as the lung with scarring and loss of lung function, as the pancreas becomes more and more damaged, a genetic cure will not reverse those things. It will restore CFTR function, but if the tissue is so damaged that it can't do its normal job, then it's still going to be not a normal tissue. And you may still need pancreatic replacement enzymes, you may still need to take mucolytics, or you may need to take some type of antibiotic if you get infections. We think, from what we've seen in the modulators, is that all of the therapies that people are taking will be less necessary. They will be, the severity of the disease will be dramatically improved. But I think it will mean different things to different people. And that will in part be determined by where they are when they start that type of therapy. So Dara, I'm sure you look at it from the view, from the lens of a parent watching your children grow and develop and live a normal, healthy life. I'll turn it over to you to kind of give your perspective as well.
Yeah, I think, obviously when my children were very young, they had very little complications. We all know this is a progressive disease, and as they get older, they have more complications. And It feels now like every time we go to our routine clinic visit, we have to add one more drug to the list, or one more treatment, or one more nebulizer. And we're constantly always adding things to our treatment plan. I think a cure for me means that we can finally stop adding so much to the day-to-day treatment, and potentially even maybe start taking some of that stuff off our plates. And that's what I really look for. I think my hope and what I think of as a cure is, just for my kids to have a normal life. It's the same as any other parent to have, grow up and go to college and get married and have children of their own. And a cure means that becomes much more possible.
I think a cure that you don't have to look at the fact that you will be dying from CF. You will live a life. There may be some constraints, but you will live a life as full as anyone else. And CF will not be the cause of the end of your life.
So that then kind of leads us to the next question, which is really a combination of two things. One is the kind of when question, right? And the other is, can you tell us a little bit about why it's so hard to predict when this is going to be, and why doing all of this science takes so long? Because I know in my experience, when I was growing up in the 90s, it seemed like it was always, 5 years. We're going to have something great in 5 years. And the 5 years didn't quite work out that way. So I understand why a lot of people don't want to put a specific timestamp on anything. But also for me, it's been really exciting to see therapies like modulators that I kind of started following when there was early science on them, and then got to see that progress through clinical trials to, a therapy that I'm taking is really pretty cool. So what do you think, Bill? When will we see something next?
Well, I wish I had a crystal ball. I wish I could look into it and say, in X number of years, we'll have the answer. What I can say is that the events that are going to drive the cure are not going to be predictable. And this is what happened in the modulator space. There were certain breakthroughs that took place, suddenly jumped us forward years in terms of progress. What we're now seeing in the genetic therapies space with some of the neurologic disorders, some of the bone marrow disorders, is that these breakthroughs are now occurring. And so we can actually start talking about a cure. I don't think 5 years ago, cure was even in our vernacular. It really wasn't something that people were thinking, oh, we could really get a one-time cure, we could have a genetic cure. So we're looking for those breakthroughs. I think it will be a stepwise process, as were the modulators. We first had Kalydeco, and then we got our Cambi. We weren't quite satisfied. We now have Trikafta. We're still not satisfied. I think what we will probably see are genetic-based therapies emerging into the clinic. RNA therapy will probably be the first because it's already in the clinic in a clinical trial, and we have some backups. There probably will be some DNA therapies that are close followers with the companies that I just mentioned that could be in clinical trials in the next 2 to 3 years. And as those results are looked at, evaluated, and better vectors, better delivery vehicles, better technologies move forward, we'll gradually see this become more and more of a reality for more and more people. So I wish I could give you a date, a time, but I can say that there has never been more excitement, because I don't want to tell you 5 years. I know that would be the mistake. But
so I'm going to kind of throw that follow-up over to Dara with kind of two specific questions. One is, I know all this, we don't want to talk about timelines, but probably about 10 people have asked, do we know where the timeline is for modulators like Trikafta for children 6 and up? So, your kid's age.
Yeah, so that is currently it's on phase 3 clinical trial right now. And we're hoping to see, the results of that very soon. I think realistically, we probably won't see anything until 2021. But you never know with the FDA. They were able to really fast track Trikafta for 12 and up. So I just remain hopeful, and I'm watching the news as like a hawk to see when that is going to come.
Yeah,
But
and the second follow-up I had, I know I said that we weren't going to talk COVID anymore, but I did want to ask, can you tell us a little bit about how COVID may or may not impact, kind of the path to a cure timeline, as well as research and studies that are going on right now?
So it's very
yeah,
clear. You want to go first, Sarah? Yeah, sure.
well, I can let you know that it is impacting research, but it varies how it's impacting research depending on what phase the study is. And I can tell you that we are still very much actively funding research right now. As you probably know, James, because you're reviewing applications for us. We have review meetings at the end of April, and we'll be making funding decisions for infection as well as other CF complications. So in terms of us funding things, that is still moving forward. Clinical trials, depending on the phase that they are some may be kind of paused until this time ends, and then they will restart. But I think, right now what we have is an opportunity to really innovate. And I think that's what we're going to see. We're going to start to see a lot of clinical trials looking at how they can maybe do virtual visits to continue progress. This too shall pass. So there may be some halting and maybe some pausing, but it will pick right back up.
That's great. So can now I ask you both to tell us a little bit about, aside from the path to a cure, how are we working to address people's CF symptoms right now? So people that are still dealing with infections and difficult to treat bacteria and all the day-to-day problems that come with CF. Bill?
So we are spending more money to develop therapies on the complications for CF than any other area of our funding portfolio. We're looking at GI complications, we're looking at liver complications, we're looking at CFRD, diabetic complications, still trying to find out better ways to understand and treat infections. This is really kind of at the top of the complications list. Dara's working with the Research Infection Initiative, and I think she will probably comment on that a little more. This is of paramount importance. And we're also funding investigators that are studying basic questions about those specific areas, trying to understand them better. So we have not given up on those. The path to a cure has not deviated our efforts in those particular areas. We think it's really important to keep people healthy and find better therapies now so that more and more people will benefit as these genetic-based therapies move forward. Absolutely critical. And Dara, you can comment on the Infection Research Initiative too.
Yeah, so in 2019, we spent $63 million on research and CF complications. And that included, like Bill mentioned, CFRD research, mental health, sexual reproductive health, sinus disease. We all know that CF is a whole body disease. And so we really do look for that research that's going to help people with all aspects of CF. You can learn more at the CFTR Isn't Just in the Lung session to talk more about where CFTR defects and how it impacts other parts of the body. And then there's also a sexual reproductive health in the era of modulator session that will talk more about how CF can affect the sexual reproductive health system.
I'm so excited that we have that session because it's something that so many people in the community really care about and have been asking about. And I think it'll be really timely for a lot of people. So tell us now a little bit about the research that's still going on into modulators. How are we learning what these highly effective modulators like Trikafta mean for people at every stage of the disease, and how we learn about things like possible long-term effects? Both, good long-term effects like having less CF progression, as well as maybe unanticipated side effects that could occur.
So I will start, but I'll talk a little bit about kind of what we're doing to really look at modulators in the laboratory, and then Dara can talk about some of the really exciting clinical trials. As I said earlier, we really want to find better modulators. We're looking in the lab to find better molecules, better combinations of molecules that are more effective at activating CFTR, that are more potent in patients, And there's a lot of work going on in that area now. But I think the essence of your question is really focused around the clinical trials that Daryl will respond to. What happens when people go on these modulators? What can we really start to expect? So Daryl, I'll turn that over to you.
Yeah, so what we learned from the clinical trials that were done by Vertex is that we know that these modulators have an effect on FEV1. People have better lung function. We know that they have less exacerbations, better quality of life. These are all things that were measured in the clinical trial. However, we don't know the effects of Trikafta on the liver and the pancreas and all of the other systems that really Vertex wasn't interested in learning about during the clinical trial. The focus was really just to get it approved. So we have one of our largest studies that the CF Foundation has funded called PROMISE, and it's actually already enrolled, and there's still some study visits happening right now, but it's really a head-to-toe look at how Trikafta is affecting a person with CF. So we're investigating the liver, we're looking at GI symptoms, we're looking at CFRD. There really is, the list goes on and on. And then on top of PROMISE, we also have other like smaller studies that are looking at sinus disease. How does sinus disease change with Trikafta? How does a woman's ability to conceive change with Trikafta? So all of this stuff, you'll learn more in these other sessions tonight. It's very exciting stuff. And then, to just build on that, because we all do want to work towards having less things, less treatments, less day-to-day burden. So we do have the Simplify study, which is really going to start to look at what can we start to remove once we get these highly effective modulators to the population.
That's great. I really love that you can give us kind of the behind the scenes look as well as being a parent. And can you, this question is going to be like, can you describe your entire job in like 1 minute? But can you give us a little bit of an idea how the CF Foundation is selecting different companies to research with and which trials and investigations to fund?
Yeah, so yes, we have like two approaches really. We have our investigators, our researchers that are at like our care centers or our institutes, and we open our research funding opportunities to them. And really, if the topic addresses something that's important to people with CF, then we are open to seeing it. So the topic area doesn't matter. So that's why we get stuff for mental health, we get CFRD, CF liver disease, we get all of the topics. And that really, that science is evaluated by external reviewers, not people from the CF Foundation, but from subject matter experts across the world. Actually, we use people in Europe as well. And if the science is good, then we will provide them funding to do their research. Companies, it's a little different because companies come to us when they're very young, startup, and the product that they have is very high risk of failure. The earlier the product is with the company, the more risk it has of actually failing and not being able to get to patients. So companies that have those high-risk products and are very early in the company's formation come to the foundation for seed investments for drugs that will help people with CF. And what that does is it de-risks, and then they're able to leverage our investments to go to other investors and build so they can move drugs to the clinic. The other thing that we do when we talk to companies, they don't obviously know a lot about CF necessarily, so we are able to help educate them on cystic fibrosis and all of the elements of the disease. We can also provide them, the specific microorganisms that people with CF are infected with. So they might not always know that Pseudomonas is a big deal or that NTM, abscessus is a big deal. So we can help educate them and we can help give them samples and help them get better tests so they can move their product along.
That's great, Dara. Thank you. And let me, again, quickly try and address kind of the big question that a lot of us with CF have is, what about the price of these therapies and the price of some of these transformational things like gene therapy? Do you have any control over that?
I'm gonna let Bill answer that.
So this is clearly an issue that we've wrestled with. In the early days when we were working with companies, if we would have required a say in the price, they never would have gone into CF with our support, and we never would have these drugs. So we recognize that it's an issue, but we really want to make sure, despite the price, that people have access to the drugs. So when we look at prioritizing our programs, we want to make sure that if we bring therapies forward that people will benefit from, that they actually are able to get those therapies. And although price is going to be part of that, we don't want price to be the limiting factor, and that's why we work with insurance companies, we work with Medicaid and Medicare offices to try to make sure that people get coverage, and we do everything we can to ensure that people get access to the compounds, which are really these life-saving new therapies. So
I'm going to have to cut you off there on that one. I wish we could talk about this for longer because it's such a tricky issue and is one that patients really feel very deeply because we're the ones, looking at these bills. But I want to get to our very last question, which is going to be kind of a quick round robin. I want us all to kind of go around and say, what is one way that every member of the community can participate in this path to the cure and help make it happen? So, Bill.
So I think there's fundraising. These are expensive therapies. Everyone in the community can help with fundraising. It's going to cost more to develop genetic-based therapies than it did for the modulators, and we don't want to run out of resources as we're going through this process. So I think that's something everyone can buy into. It's been the backbone of the foundation for the last several decades. I don't think that's going to change.
Sarah?
Well, I think really we need the voice of people with CF to be involved. So I really encourage you, if you're not already a member of Community Voice and Research Voice, to please consider joining. It really helps us to understand what are those questions, what are those problems that people with CF are really mostly concerned with. And then we are able to really put a call out to our researchers to get that research to us. So we have many different ways that you can be involved. Your level of involvement can range. If you want to take a break and not be involved for a time, we don't go off of the list. You have an option to be as involved as you want. So
that's great. Yeah, I love being part of Research Voice and Community Voice, and that's how I got here.
Yeah.
And for me, I'm going to say it's going to be all of us patients with CF participating in clinical trials and saying, yes, I want to help study CF, and yes, you can use my clinical data and my samples, and please learn as much as you can from me. And it makes me think of something that one of our speakers, Zan Novakovsky, said during our planning process, which was essentially that if we don't show up for the science, then the scientists can't figure out how to make us better. And I thought that just really brought home how important it is to have patients participating in all parts of the clinical trial process, from understanding what we should research, like you were talking about, Dara, to actually doing the research and participating in it. So that is really all the time that we have for our discussion. I wish we could keep going for the rest of the evening, but we've got lots of exciting other things for us to do here at ResearchCon. I do want to mention, for anyone that might have closed out of the event homepage, you will be able to get back to that by going to the link posted in the chat or across your screen. So to get to the next part of ResearchCon, you will go back to that first event page that you saw. And now I would like to welcome John back to close us out and kind of tell us.
Thank you, James. And thank you, Dara and Bill. That was a really wonderful keynote and discussion. I think we all learned a lot. So as James mentioned, we'll now be transitioning over to the information sessions. So the first round of these information sessions will be starting at 8:15 Eastern. So that gives you a little under 15 minutes for a break. So grab a snack, use the restroom, and then come back. I do want to remind everyone that there are a variety of resources related to all the event sessions in the resource room. That's been mentioned a few times already. So you can access that from the homepage of the event, where you'll find the keynote slides and other items our speakers thought you might find valuable. So please spend some time checking out those resources and download anything that you might want to keep. So now you can close the window and return to the ResearchCon environment. So thanks a lot for coming to the keynote, and we'll see you in the information sessions. Thanks, everyone.